An estimated 3,500 Australian men will die from prostate cancer in 2017, with a further 16,500 new cases being diagnosed. Prostate cancer is unregulated cell proliferation localised in the prostate, which has the potential to metastasize primarily to the lymph nodes, the bone, the lungs, and the brain. As defined by Hanahan and Weinberg in 2011, there are 10 hallmarks of cancer that are acquired by cancer cells as they progress towards human tumorigenesis. Therefore, treatments targeting these hallmarks aim to disrupt fundamental components of cancer development. Our team of esteemed researchers at the University of Sydney have developed a new treatment targeting the cancer hallmark of resisting cell death. This will be done by upregulating protein phosphatase 2A, which in turn stimulates apoptosis. In doing so, we provide a novel treatment designed to reignite cancer cells' own self-destruct mechanism that has great potential, not only on its own, but also in combination with chemo and radiotherapies.
The intrinsic apoptotic pathway is a mechanism inherent in all cells, which upon activation causes controlled destruction of the cell and the recycling of its components. However, the regulation of the mechanism is altered in prostate cancer cells, so that the system is largely inhibited. This results in cancer cells that do not undergo regulated cell death, and instead continue to proliferate and become increasingly difficult to treat therapeutically.
A family of pro-apoptotic and anti-apoptotic proteins, known as the BCL2 family, regulates the intrinsic apoptotic pathway. Our target, protein phosphatase 2A, is an upstream regulator of this pathway. Apoptosis is caused by the permeabilization of the outer mitochondrial membrane. This permeabilization is triggered by two main pro-apoptotic proteins, BAX and BAC, which oligodimerize at the membrane, forming pores, which results in the efflux of several proteins and macromolecules, most notably cytochrome C. Cytochrome C activates a cascade of caspases that induces a variety of cellular changes within the cell, leading to apoptosis. Anti-apoptotic proteins regulate this system, in particular BCL2, which dimerizes with BAX, inhibiting it from forming pores in the outer mitochondrial membrane, and thus inhibits apoptosis. In prostate cancer, there are a variety of reasons why the regulation of this system is malfunctioning. It may be due to upregulation of anti-apoptotic proteins, or the downregulation of pro-apoptotic proteins. Our treatment aims to reduce the activity of BCL2 in cancer cells, and thus increase the activity of the pro-apoptotic protein BAX. We will achieve this by upregulating active protein phosphatase 2A. Protein phosphatase 2A is an endogenous regulator of the intrinsic apoptotic pathway and is responsible for the dephosphorylation of BCL2-associated death promoter, or BAD. Upon dephosphorylation, BAD is able to bind to BCL2 and inhibit it from binding to BAX. This leads to an increase in active BAX at the outer mitochondrial membrane, and therefore the activation of the intrinsic apoptotic pathway.
Our treatment will use a lentiviral vector to upregulate protein phosphatase 2A within the prostate cancer cells. The advantage of using lentiviral vectors is that we can increase transcription of a desired target within a specific cell type. In terms of our treatment, we will target the prostate cancer cells specifically and then incorporate the viral vector into the cell nucleus. Prostate-specific membrane antigen, or PSMA, is a membrane-bound antigen found in a number of cells. However, it is shown to be substantially upregulated on the surface of prostate cancer cells. Our treatment will utilize a PSMA-specific antibody that binds at a certain efficacy to PSMA on the surface of prostate cancer cells. Following this, our viral vector will translocate to the nucleus and in turn upregulate transcription of protein phosphatase 2A.
As with all novel treatments, there are inevitably limitations. A downside of using a lentiviral vector is the potential for immune rejection. This can be overcome with co-administration of immunosuppressants, as is the case in many other treatments. Also, in upregulating protein phosphatase 2A, even just within the prostate cancer cells, there remains the potential that this could cause unforeseen consequences. If this is the case, clinical trials will make these apparent. However, this novel therapy shows great promise due to its potential to supplement existing therapies, as well as the ability to avoid off-target effects through the specificity of the lentivirus to the cancer cells.